everyone, we're Team 26. Our topic today is antibody drug conjugates for cancer therapy. What is an antibody drug conjugate? An antibody drug conjugate, or ADC, consists of an antibody that is covalently connected to a cytotoxic molecule called the payload by a chemical linker. Antibodies function in our immune system to detect and neutralize pathogens by targeting antigens on cell surfaces. Based on the phrase antibody drug conjugates, you might ask, so what's the purpose of conjugating an antibody to a drug? If we take one step back to look at cancer therapy, specifically chemotherapy, cytotoxic agents are used in an effort to kill cancer cells. Unfortunately, they are nonspecific and target all types of cells in the body, causing side effects such as hair loss, fatigue, and infection. The lack of specificity is the main cause of these side effects. The purpose of conjugating antibodies to these cytotoxic drugs is to allow the antibody to help guide the drug to only cancer cells and not the healthy ones. In this diagram, the pink Y shape represents a monoclonal antibody selected to bind to a specific antigen. Monoclonal antibodies are antibodies that all bind to the same epitope, and the specific kind is selected such that it binds to an internalizing tumor-associated antigen that is minimally expressed on normal tissues. As we can see here, a cancer cell surface contains special cancer-associated antigens that normal cells do not have. The special antigen can act as the target for antibody binding. The black line represents the linker region by which the cytotoxic molecule, or payload, shown as a red circle, is attached to the heavy chain of an antibody at a conjugation site by a covalent bond. The conjugation site is represented by a green circle. The linker must be carefully engineered to be stable in extracellular conditions to minimize the systemic release of the cytotoxic payload, but be rapidly cleaved by intracellular proteases. If the linker was not stable in extracellular conditions, there would be no advantage over traditional chemotherapy. Here's a general overview of the mechanism by which ADCs work. First, the ADC is administered via intravenous infusion directly into the bloodstream, where it will find and bind to specific cancer antigens causing internalization. The orchestration of this step is more difficult to engineer than it appears on paper, and the actual uptake of ADCs is extremely low. Any successfully uptaken ADCs enter the endocytic pathway and go to the lysosome, where the linker is digested by lysosomal enzymes, releasing the cytotoxic payload. Additionally, the disulfide bonds that connect the heavy chain and the light chain of the antibody cannot be sustained at low lysosomal pH. So, in the lysosome, both the antibody and the linker are degraded, and the cytotoxic agent is released. The released cytotoxic agent is active and will bind with the microtubule or DNA to cause disruption. The disruption of DNA, even within a cancer cell, will eventually cause cell death. From the description of ADC's mechanism, the biggest advantage of this method towards cancer is obvious: specificity. Since this kind of therapy delivers the cytotoxic agent directly to the cancer cell aggregations, it makes the treatment much safer. Even though we cannot say that there is no side effect associated with ADCs, it is definitely less than the traditional chemotherapy.